Cells have a variety of ways of using chemicals to communicate with each other. An important means of intercellular communication in the nervous system is synaptic transmission, in which one neuron releases neurotransmitter molecules onto another. The neurotransmitter molecules bind to and activate receptors on the target cell. The activated receptors then activate target molecules within the cell. Other receptors act as ion channels that transduce the chemical signal into an electrical signal. Other well-characterized forms of chemical communication include paracrine signaling. In paracrine signaling, a cell releases signaling molecules that activate nearby target cells with appropriate receptors. Paracrine signaling acts over a longer distance than synaptic transmission. Another form of chemical communication, called endocrine signaling, refers to the secretion of hormones into the bloodstream. The hormones thus reach tissues throughout the body. Those cells with appropriate receptors become activated. Chemical signaling of any sort requires several components: a molecular signal that transmits information from one cell to another, a receptor molecule that transduces the information provided by the signal, and a target molecule that mediates the cellular response. The part of this process that takes place within the confines of the target cell is called intracellular signal transduction. Within the cell, the signal is typically amplified. Let's look at an example of signal transduction, beginning with a receptor protein that can bind to the neurotransmitter norepinephrine. The activation of a single receptor by norepinephrine can lead to the activation of numerous G proteins inside cells. This is the first amplification step. These activated proteins can bind to other signaling molecules, such as the enzyme adenylyl cyclase. In this way, adenylyl cyclase becomes activated. Each activated enzyme generates a large number of cyclic AMP molecules. This is the second amplification step. Cyclic AMP binds to and activates another family of enzymes, the protein kinases, that can phosphorylate many target proteins. While not every step in this signaling pathway involves amplification, overall, the cascade results in a tremendous increase in the potency of the initial signal.